Yeah, no worries.
Okay. Yeah, people tend to trickle into these seminars a little bit late, but I'll just give it a couple more minutes. Okay, I'm going to go ahead and get started with the introduction. Today we have Nicholas Heaton as our seminar speaker for the Immunobiology seminar series. Uh, Nicholas will talk about tissue-specific interferon regulation and the effects on influenza virus pathogenesis. So Nick is an assistant professor at Duke University School of Medicine, and he's been studying viruses and the immune system for a long time. Since graduate school at the University of Chicago, his thesis defined really new mechanisms for how viruses modulate cellular lipid metabolism. And then Nick did his postdoctoral work at Mount Sinai, where he worked with Peter Police on the mechanisms of influenza virus pathogenesis. I've known Nick for several years now. I've been a big fan of his work. We've attended these annual conferences together, and I'm really excited to hear about his new work in this field. At Duke, his group really has worked on three major areas, all involving viruses in the immune system. He's really taken advantage of recombinant viral technology to understand how infection can alter basic cell physiology. And with this, he's also defining the host factors that are required or restrict respiratory viral infection. And he's also using this technology to develop improved vaccines, something that's highly relevant to our current situation. So without further ado, I'll have Nick take over. And thanks, Nick, for giving this presentation.
Yeah, absolutely. Thank you, Jack, for the introduction and for the invitation. It's a pleasure to be with you today, if only virtually, and tell you a bit about the work that we've been doing recently, really trying to understand the regulation of interferon, and in particular the negative regulation of this process, and understanding how that affects the diseases that influenza viruses cause. So I'm going to give you a one-slide introduction to influenza viruses. These are segmented negative sense RNA viruses, and an electron micrograph is shown on the top left. A cartoon is shown on the top right. Essentially, all that means is that the genomic material is encoded in RNA and across 8 different genomic segments, and these RNAs have to be copied before they can be translated. Members of this family include the influenza viruses, among some others. Influenza A virus is unique. It has really a large host range, notably birds, in addition to humans and other mammals. When we talk about flu, what we're really talking about is influenza A and B, at least with respect to human infection. And even though we perform seasonal vaccination campaigns, have at least 3 classes of small molecule inhibitors, these viruses still cause significant burden on human health. And prior to the current pandemic, I would have said the most kind of medically relevant respiratory virus currently affecting people. So what I want to do in terms of framing the talk is get you thinking about cellular fate after viral infection. This is really kind of our entry point into the studies that I'll tell you about today. And I'll start with a concept that I think is intuitive to most people, and that is, depending on the virus and sometimes the specific type of cell, the infection of that cell can lead to different outcomes. So for example, hepatitis C virus infects hepatocytes, and when it does so, there's an active persistent replication. The cells aren't killed, the immune system doesn't kill the infected cells, the virus just continues to replicate at a low level in these cells indefinitely. That's in contrast to something like a herpes virus. Herpes simplex virus, for example, if it infects a neuron, will undergo a transition to latency, where the virus will still be there, but very few viral proteins or transcripts will be expressed at any given time. And a third option is probably what most people think about when they think about viral infections, and this is a cytopathic infection. That means the infected cell is going to die. For example, rabies virus does this. So what about flu? I promised you that the talk was going to be about flu, and really there's 2 cell fates that the field has appreciated as being relevant to influenza pathogenesis. The first I'm diagramming here on the left, this is a non-permissive cell type, or an abortive infection. So there are certain cells in the body, for a variety of reasons, that when the virus binds its receptor, it either can't enter or enters and finds a non-permissive environment. Basically, this virus never replicates, and this cell then as a result is fine. Nothing happened to it, it persists in the host and continues to perform its kind of physiological roles. The second, which is kind of where the action happens, is when the virus initiates infection in a permissive cell. The virus will replicate itself, copies of the virus will spread to neighboring cells. And the idea is that flu is a cytopathic virus similar to rabies. The cell can die a variety of ways. It can either be killed lytically, necrotically, or the cell can undergo apoptosis. Or as you can imagine, these cells are full of virus antigen, and the immune system efficiently comes through and eliminates those cells from the body. And really, these 2 classes of cell fates are how influenza virus pathogenesis has been appreciated historically. This is kind of the lens through which the mechanisms of virus disease have been viewed. And the way that these projects that I'm going to tell you about today really got started was wondering, is this it, right? Is there anything else that can happen? And in particular, we envisioned a potential third scenario, which I've diagrammed here, called permissive and surviving. What if there were cells, epithelial cells here, that got infected, and the virus replicated, actively expressed all its proteins, even completed its replication cycle? But then the cell won. Instead of going down this pathway, the cell beats the virus, clears the virus, eliminates all traces from itself. It could presumably persist in the host, and we wouldn't know because, to a large extent, this cell looks like this cell. And for a variety of historical reasons, get interested in the inflammation that infected cells cause, and one could envision a scenario where both this cell and this cell, having experienced virus at some point, could be contributors to kind of this inflammatory state in which the virus replicates. So this was the question: can we provide any evidence that this third fate may indeed be occurring? And as I alluded to briefly before, this poses a technical kind of labeling issue, if you will, because of how we detect virus. So we detect virus like everybody else does. We look for viral nucleic acid, we look for viral protein, and this cell has it, right? So we can tell the difference between this cell and these 2. But these 2 cells, look the same to us now that the virus has been cleared. So in order to look for this third cell fate, we essentially needed a way to permanently label any cell that had ever experienced virus replication. And the way my lab approaches many questions is molecular virology approach, really a virus engineering approach. Can we build viruses that allow us to solve these problems? And in this case, we generated a virus that expresses the bacteriophage protein Cre recombinase. And the way this solves our labeling issue is shown here on the right. So the virus is going to express Cre. Cre is not packaged in the virion. The virus requires active replication to express Cre, which by itself doesn't do any labeling. But if the cell contains the appropriate reporter cassette, we can get labeling. So the way this works is a constitutively active promoter is followed by a transcriptional stop cassette. So RNA polymerase gets on here and goes to here and falls off. But flanking this stop cassette are these LOXP motifs, which are the DNA sequences that Cre recombinase recognizes. So you're not getting any signal made when the virus infects cells. Cre recombinase recognizes these sites and removes the DNA in between them. What that accomplishes is now the reporter is put under control of this promoter. In this case, a red fluorescent protein. The cell becomes red. And the important thing is here, this is in the host cell, right? So the virus turns this on and this process is irreversible. But if the virus is subsequently cleared, this cell stays red, right? So now we can tell the difference between our two cell populations. If a virus has never been infected, it's not red. If it's actively infected, it's full of viral protein, which we can detect with antibodies or anything else. And if it was infected but cleared the virus, it's fluorescently red. So this is the system. Seems like we should be able to ask our question. And indeed, we can. So we made this in an H1N1 background. There's a mouse line that expresses that exact cassette that I just diagrammed for you. We simply infect these mice, look over a time course and say, do any cells survive? This is a representative flow cytometry plot. You can see prior to infection at day 0, there's no red cells. At day 5, this is a time point of active virus infection. Viral titer is essentially peaking or just starting to come down by day 5. You can see there's a bunch of red cells. The virus is cleared in this system by day 10. We have a bunch of red cells. We can go to day 21, there's still red cells. We can take these animals out to the end of their natural lives. There are red cells that persist indefinitely. So it seems like cells are surviving. This is the same flow plot. This is viral titer from the lungs. The virus is really cleared rapidly as soon as the adaptive immune response comes online. And this is what it looks like. So this is the trachea. This is a tracheal hole mouth. What we've done, the trachea is a tube. We've taken this tube and cut it lengthwise and laid it flat. So you're looking like you're in the airway at the site of the trachea. And you can see that we have this whole population of cells. Up to 5% of the apical epithelia after a flu infection actually experienced active virus replication and is now persisting in the host. So this represents a completely new population of cells, a population of cells that we didn't think existed. We would say at this point that none of the cells historically by day 21 experienced any virus infection. They would have been killed by the virus of the immune system. And now we know we have a large population of cells that in fact did interact with the virus in a meaningful way. So the next question for us is what happens to these cells? Typically, how are these cells behaving? We can envision two possibilities. The first is that the cell just shakes it off, forgets about it, and goes back to doing what it was doing beforehand. The second option is that something about this infection changed that cell. And now you have a phenotypically distinct population of cells in the lung. A virally induced population of cells, if you will. So again, we want to distinguish between these two possibilities. The way we looked at that was to perform a transcriptional profiling experiment. And so I'm showing you here the viral RNA. We took cells at day 0, 2, 5, 7, 14, and 24 post-infection. You can see viral RNA at early time points. The cell is full of viral RNA. It's 2 or 3% of the total transcripts of the cell are viral. Then that's cleared by this time point, and by day 14, viral detectable in the cells. But what we care about here is the host response, and so we can sort the transcriptional response to the virus by genes that are most upregulated at day 2, when the virus is really replicating robustly in these cells. And I'm not showing you the genes here, but this is everything you would expect. These are antiviral genes. These are cell stress response genes. And essentially, as the virus is cleared, these profiles turn off. But what we wanted to know is if something weird happened here after the virus was cleared, so we can resort this data set by genes that are upregulated late. And what I hope you can appreciate here is that genes are turned on. The cell really acquires a discrete transcriptional profile from either prior to infection or during active infection upon the resolution of infection. So we took this to mean that it was option 2, that these cells are changed. They're reprogrammed by the virus in some way to become something new. And transcriptionally, they're different. Presumably phenotypically, they're different. This is something we were really interested in, and something that we've worked on for a number of years, and we've published several papers on these phenomena. I'm not going to take you through them because they're public, but suffice to say, these cells can influence susceptibility to secondary infection. Just their ability to survive and persist in the lung helps maintain barrier function. But the phenotype that I want to highlight is from one of our first papers describing this phenomena. And the takeaway is that these survivor cells, upon clearing the virus, still maintain this hyperinflammatory transcriptional phenotype. So they're continuing to make inflammatory mediators that is contributing to the immunopathology associated with flu infection. And the way that we know this is we did an experiment, which is a kind of a variation on what I showed you before. So instead of using our fluorescent reporter mouse to follow these cells, we can use a mouse that induces the expression of the diphtheria toxin receptor. So murine cells don't express the diphtheria toxin receptor. In this case, our survivor cells now specifically are labeled with this toxin receptor. We can go in and apply the toxin to the animal and specifically deplete just those cells that survive. So we can set up a condition where we have 2 animals that have both been infected with flu, both survived. In one case, we've depleted all the survivor cells, just like if the virus would have killed everything. And in the other case, allow these survivor cells to persist. We saw an interesting and counterintuitive phenotype. So these are our bronchioles. This is H&E stain of the lung. This is the pseudostratified epithelia, where the virus really likes to replicate. And this is what it's supposed to look like. This is from a naive mouse. This is a wild type mouse that has its survivor cells. And you can see extensive damage of the epithelia here. These cells, which are normally columnar and packed in there, most of them are dead. And the ones that are still there have laid flat, trying to just cover this basement membrane and protect the integrity of the airway. Now, this condition on the right is the toxin treated. So we've actually killed a bunch of cells here, a bunch more cells, but you can see that the epithelium looks much better. And again, it's published, so I won't show you the details, but it turns out the takeaway is that it's the inflammation that these survivor cells that are making, they're bringing in more immune cells, particularly macrophages and neutrophils, that are continuing to cause damage. And of kind of all the inflammatory mediators that one could think of, it turns out that it's the interferon-induced cytokine and chemokines these survivor cells are making that are contributing to this phenotype. Okay, so that's what we know. And then the question for us became, why is there active interferon-induced signaling in these cells? We know a lot about interferon signaling. This is an important kind of part of the canonical antiviral response. When a cell detects virus, it makes interferons. Here I'm showing you type 1 interferon, that then binds through the interferon receptor. This is JAK-STAT signaling, binds ISRE elements in the promoters of genes, of specific genes called interferon stimulated genes. And this mediates this antiviral state. This is the pathway that is turned on in our survivor cells. We think is contributing to this immunopathology phenotype that we want to limit. But it's doing so at a time point well past virus clearance. There's no interferon anymore. So why are these genes being expressed? Our initial thought was that there's some dysfunction in the downregulation of this pathway. You can look at this via looking at phosphorylation of STAT proteins, for example. So we go through it systematically. And everything that we know, everything that the field knows of, we looked at, and we can't find any problems. Everything looks normal. The expression of these genes is appropriate. The post-translational modifications like phosphorylation is all appropriate. It seems like this pathway should be turned off, but it's not. So we interpret this as there are additional regulators of interferon signaling outside of what's diagrammed here. We need to identify to understand what's making these survivor cells inflammatory. So we want to screen for genes, new proteins that regulate this process. The first thing that we need is a reporter system, an interferon reporter system. So we generated a reporter construct. It has a number of these interferon stimulated response elements in the promoter. And basically, long story short, this promoter is not active until interferon signaling occurs. You make a GFP variant, which folds and fluoresces very quickly, and then is also degraded very quickly. And it works. We can add interferon, and within 6 hours, all the cells are bright green. So we have a system now where we can screen for interferon regulators. We want to do high throughput screens, and the approach that we took was CRISPR screens. I'm sure all of you are aware of CRISPR-Cas9 technology and how it can be used in screening. Just briefly walk you through it. So on the left is kind of what you think of as canonical Cas9 technology. Cas9 is a protein which induces a double-stranded break. It's targeted to a specific locus via the specific sequence of an sgRNA. If you want to knock out genes, you target Cas9 to one of the exons. It causes a DNA break. It's repaired inappropriately, typically causing a frame shift. And if you were to do a screen with such technology, you would identify the factors necessary for interferon regulation, factors that when knocked out compromise interferon regulation. But there's a variant of Cas9 technology that can be used to activate genes. So now it's dCas9, it's catalytically inactive Cas9. So Cas9 can't cut DNA anymore. It's still targeted to whatever locus you want by virtue of homology of the guide RNA sequence to your target, but now it gets targeted to promoters. And Cas9, via a variety of mechanisms, also recruits a bunch of transcriptional activators, so you can drive gene expression instead of knocking genes out. And if one wants to do a screen with such technology, you would find the factors that were sufficient for interferon regulation, right? The factors, when overexpressed by themselves, can control this process. So we decided to take the latter approach. It's kind of the most simple model, right? If there's one thing that's changed in survivor cells that can explain the phenotype. So anyway, we decided to start here. The screen we did is diagrammed as follows. So we take a large number of cells, which have all the pieces for this Cas9 activation technology and the interferon response reporter. And then we transduce them using lentiviruses with a population of guide RNAs. We do this in such a way that cells either get 0 or 1 guide RNA. So we're only turning on one gene at a time. We allow enough time for this Cas9-based technology to turn on gene expression. We collect some of the cells as an unselected input. The rest we treat with interferon. The vast majority of the genes that have now increased in expression don't do anything for the interferon response. we collect them for FACS and they're green, right? These cells turn green when they are exposed to interferon. So most of them are here, but some of them had a gene turned on that made them no longer responsive to type 1 interferon. That's the population we collect, sequence both of those, and perform bioinformatics to say what is the guide RNAs and what genes are targeted by those guide RNAs, which are really enriched in this population. So I don't have the time to go through all the details of that analysis, but at the end of the day, we got 3 genes: C1 GALT1, ETV7, and NUP153. And we can clone these genes and overexpress them to control for off-target effects that Cas9 may be having. And we see in all cases with our interferon response reporter, when we overexpress these genes, we get less interferon response. We can do a control just with a plasma that expresses GFP. And we see that C1 GALT1 suppresses the response a little bit, so it's not fully specific for the interferon-induced expression of this GFP. But these two look good. And we decided to focus on ETV7 because it has not been previously reported to affect interferon signaling, but this gene itself is a strong ISG, so it's turned on after interferon signaling occurs. It's also a member of a family of well-characterized DNA binding transcription factors. This is seeming like a gene or a protein that could really be mediating this type of interferon regulation that we're looking for. But this is just a reporter, right? What about real ISGs? So we can overexpress it and perform qPCR for representative ISGs. Here, ISG15, IFIT1, you can see overexpression of the gene compromises the induction of that gene, at least at the transcript level. At the protein level, everything looks good. IFIT1, control cells that are overexpressing mCherry. The gene is turned on when you add interferon. Accumulation is reduced, as well as some others. So it seems like it's good by itself. Overexpression of this gene is sufficient to suppress the interferon response. It turns out that the reciprocal is also true. If we knock out this gene, there's physiological relevance for its activity. Now, when you knock out this negative regulator, the expression of interferon stimulated genes goes up at the RNA level and at the protein level compared to control treated with interferon. You can see that the accumulation of these ISGs is much more rapid. So again, at least for us, it's hard to know what, this amount of change at the RNA or at the Western blot level means. But I told you that the interferon response is designed to control viruses, right? So one way to assess, kind of the importance of these changes, of the physiological relevance of these changes, is to see if we can affect control of virus replication. So again, the model is that ETV7 is a negative regulator of ISGs, which control virus replication. So if we knock it out, ISG expression goes up, antiviral activity goes up. We expect if we infect these cells, virus should replicate poorly. And it turns out that it does. So in the control, the virus, this is with influenza A virus grows well, and virus replication is controlled in the knockout, where you're getting basically hyperactive interferon response. So this all seems good. This seems, like the protein can do it with real genes in a real context, and the magnitude of these changes is relevant. Next question is, how is it acting? So again, it's a DNA binding transcription factor. We know all ISGs are turned on by virtue of having this ISRE consensus motif, which I'm showing you here. And it turns out that the entire family that ETV7 is in has had its binding motif characterized. And this is the motif for ETV7. So what really matters is in the middle, this GGAA motif. So knowing this, if we look at the ISRE consensus saying, potentially does ETV7 bind these motifs and outcompete the ISGF3 complex, which is what drives transcription? We see 2 sites that could be really good ETV7 binding sites or not, depending on what this N is, right? If it's a G, it's a perfect consensus site for ETV7. And if something else, ETV7 presumably won't bind. So that's the model. And if we look at ISREs, right? I mean, there's actual ISREs and the promoters of all these ISGs. I'm just showing you a subset here. Do any of them have motifs that would potentially be bound by ETV7? And like many things, the answer is that it's complicated. Some genes do not. IRF9 is an ISG. Its ISREs do not have motifs to be bound by ETV7. Some have multiple ETV7 binding sites in one SRE. Many have at least one. The takeaway here is it seems like this is a likely mechanism for how ETV7 could work, but also it predicts that different ISGs may be differentially affected by ETV7. You can have a ISRE and either be a consensus or not for ETV7. So predicts potentially that there's differential effects across ISGs. And so we can just sequence cells treated with interferon and see if this is the case. And what I'm showing you here is a heat map in cells expressing M-cherry or ETV7 with or without interferon. The control cells, when you treat with interferon, all the ISGs go up. That's yellow in this graph. When you have ETV7, there's globally a suppression. But this condition doesn't look like this condition. There are some ISGs that are really strongly suppressed and some that are almost unaffected, despite the fact that ETV7 is being overexpressed there. We can genetically test our hypothesis that it's because of those binding sites. It turns out that the initial ISREs in our reporter, all 7 of them that are in that site, have consensus ETV7 binding sites, which makes sense why we pulled it out of our screen. We can mutate those to still be ISREs, but not be binding sites for ETV7. And when we do that, we lose sensitivity to the expression of the protein. So again, the parental, which has these sites, ETV7 expression, suppresses expression from the reporter, whereas the mutated construct here now still responds to interferon, but no longer has any sensitivity to ETV7. We can biochemically show that with a pull down as well. Biotinylated oligos corresponding to regions in the promoter. Here we're using one from ISG 15, which has this consensus motif. We can mutate one position so it's still an ISRE, but not a predicted good substrate for ETV7. We get differential pull down. The wild type oligo pulls down ETV7 just fine. This doesn't pull it down at all. So all of this data together leads us to this model, which breaks ISG expression into 2 classes, at least with respect to ETV7. So interferon binds a cell. You initiate this JAK-STAT signaling. And at the beginning, this complex, ISGF3, which is kind of the ultimate downstream signal after interferon signaling, it binds ISREs, which may or may not be bound by ETV7 eventually. At the beginning, it doesn't matter because there's no ETV7. I told you ETV7 itself is an ISG. So early on, you make all ISGs the same. But as ETV7 accumulates, it goes back into the nucleus and now suppresses specifically those ISREs. It binds and suppresses transcription of those ISREs, which have the ETV7 motif, which is called an EICE. And we think that this globally probably acts as a brake to control interferon signaling from getting out of control, if you will, but also likely contributes to the temporal expression and regulation of ISGs over time. Okay, so we got into this. We were looking for negative regulators of interferon signaling. We found what we think is a new one. We think we know how it works. But again, we got into that because we cared about survivor cells and why these cells are continuing to express ISGs even though the interferon ligand for that system is gone. I told you all the things that we know about in terms of canonical interferon signaling was not dysregulated, but when we look at ETV7 in these cells, we see it's dysregulated. In particular, ETV7 is not being expressed in survivor cells where the other ISGs are. So what we can do is now restore ETV7 expression in our survivor cells and say out of this profile that we already know happens in survivor cells, how much of this goes away now when ETV7 is present. And the idea is that ETV7 is potentially something downstream of the process of cells surviving themselves that we may be able to target, right? So in a person infected with flu, after clearance of the virus, we want people to look like this and not this. We can make this happen by genetically depleting the cells in experimentally infected animals. That's obviously not a good approach for people. We don't want to be trying to eliminate cell populations from people. Something we think is a more compelling idea is targeting the mechanisms in these survivor cells which are leading to this inflammation. ETV7 likely being one of them, to either limit the initial damage that's done or potentially enhance the rates of lung regeneration. So all of this is in the lung, and as influenza virologists, we study the lung because this is where viral replication occurs. The virus stays in the respiratory tract with the exception of some highly pathogenic strains of flu, not because viral receptor expression is limited to the lungs, but because the virus requires a protease to be infectious, and that's expressed only in the respiratory tract. So this is where we study the disease process, but the inflammatory cytokines and chemokines that we were just talking about are not restricted to the respiratory tract. This is a heat map from a paper done by one of our collaborators from an experimental human infection model. They're taking peripheral blood from people's arms hours after they're inoculated with a virus. And so in peripheral blood, you can see there's this strong kind of inflammatory signature. These cytokines are being made in the lungs, but then they're circulating across the entire body, including our friend interferon alpha, which is type 1 interferon. So we started thinking, although we've been lung focused, and this is certainly where the virus is inducing these processes, maybe we need to look across the body to understand kind of the full manifestations of virus disease. Mechanistically, and this is known, so flu, the disease, influenza, caused by the virus, is not restricted to the respiratory tract, right? There are systemic things like fever, there's headache, joint pain, muscle pain, a lot of GI manifestations as well. We typically don't study this because it's not the lungs. But we thought this makes sense that, if we're interested in the disease is systemic. And now that we're in this whole kind of inflammatory signaling, what are these cytokines doing? How is it regulated space? Maybe we should look at non-lung sites of inflammatory signaling. So what I'm talking about is context-specific interferon signaling, really. And what I mean by that is that we know that some cells and tissues can tolerate inflammatory interferon signaling just fine, like our survivor epithelial cells. These guys start expressing ISGs, and they're particularly, they don't care. They do that indefinitely. Many cells and tissues tolerate transient interferon signaling. That's how the system is supposed to work. A virus is there, make interferon, it puts cells into a restrictive state with respect to viral infection. The virus is cleared, it goes away. But some cells and tissues are highly sensitive to interferon signaling. They can't tolerate any amount at all. So what are these tissues? Because these are the lowest hanging fruits, right, for us to go after. These are the ones that we would expect to be most affected by the interferon made in the lungs during virus infection that is then circulating. It's where we think we are most likely to see a phenotype. So what should we go after? Well, it turns out that fetal tissue is exquisitely sensitive to interferon signaling. We know this via a lot of work on Zika virus. This is a paper from Akiko Iwasaki's lab. Show that type 1 interferons, same interferons that we were just telling you about in the regulation of whose signaling I was just telling you about in the lung, these cytokines are actually major drivers of fetal demise after Zika virus infection. And there's a paper from Oliver Schwartz which showed more about why this interferon signaling is so bad for the developing fetus. But I want to show you one panel from a Kiko's paper, which I think drives this home. So again, this is a Zika virus model. And the animals are crossed in such a way that some pups either have the interferon receptor or they don't. So in the same pregnancy, same infection, you have pups with different genotypes, and these ones that have the interferon receptor can respond to interferon, these ones can't. They're infected with Zika. Zika is replicating in these fetal tissues. And what's really striking, I think, is that the interferon knockouts, the developing fetuses are phenotypically fine. Viral titer goes up. There's actually more virus here because interferon is disabled. There's no inflammatory signaling happening. So even though there's more virus, the fetuses are still fine compared to the interferon competent animals. When you have this inflammation, you get this dramatic fetal reabsorption phenotype. So this argues, at least in the mouse model, that the cytopathic effects of the virus killing cells in the developing fetus, is not as detrimental as the inflammatory signaling that the virus induces. But thinking back to what we know about flu, this causes a paradox, right? Because we know that flu infection causes systemic interferon. Happens in pregnancy and not in pregnancy. But unlike Zika, flu infection during pregnancy is not associated with severe fetal developmental outcomes. There are some kind of negative outcomes like preterm delivery or low birth weight, but nothing dramatic like this microcephaly phenotype. So in both cases, we have interferon, but why doesn't something bad happen with flu or something worse happen with flu? So to restate that with a cartoon, here's the idea. There's differential interferon signaling requirements between maternal tissues and fetal tissues. When there is a maternal viral infection in the lungs, for example, an active antiviral response here is required to limit maternal morbidity. But at the same time, this interferon is traveling, this inflammation is traveling systemically, and the response here must be avoided. So how does that happen, right? And we're now on this negative regulation of interferon idea. And the question is there something specific that suppresses interferon signaling here, but allows it here to resolve this kind of issue that we're facing? Again, if we look in the literature, or if we look in the hits on our previous screen, nothing is a great candidate for this differential control of interferon signaling. So we decided to do another screen. We're looking now for genes that are necessary for the shutoff of interferon signaling, which we think is more consistent with this model this paradox we're trying to answer. We're using the same interferon reporter system where interferon makes cells turn green, but now we've optimized conditions where we can wash out that interferon and watch these cells lose their fluorescence again. Again, in flow cytometry, this can happen. Prior to treatment is red, with treatment in blue, and then after washout, the cells lose their fluorescence again, that's in green. So the screen that we designed to look for more regulators, kind of in this different context, is shown here, and this is a knockout screen. So again, we have a number of cells. Again, we introduce one guide RNA per cell. We collect some as an input. And then here we treat cells with interferon, and all the cells turn green. And then we wash out the interferon, and we wait for the cells to all lose their fluorescence. Then we collect them, and what we find is a population of cells that are still green. While most of them have turned off, there are some cells that are still green. These are cells that have lost something required for them to suppress the interferon response or turn it off. We then do sequencing of kind of the post-selection and pre-selection populations. And this is a representation of that data. So there are a bunch of non-targeting guide RNAs that don't do anything in the screen. We can use that as a cutoff for a real signal. We pull out, you can't see them all because they're on top of each other, but we pull out essentially all the components of the proteasome, which is a good positive control for our screen. The proteasome is required to degrade that GFP signal. So when we get rid of pieces of the proteasome, it can't get rid of GFP, these cells stay green. It doesn't have anything to do with interferon regulation per se, but it shows that our screen worked. And out of all these hits, what we became very interested in was this protein GPR1. So GPR1 is a G protein coupled receptor. In particular, its ligand that activates its activity is 17-beta estradiol, which is a component of estrogen. This dysregulation is associated with cancers, and we thought it was potentially a good candidate for regulating interferon responses during pregnancy because, as I'm sure you all are aware, estrogen levels increase dramatically over the course of pregnancy. And now we have a predicted interferon regulator that is whose activity is potentially activated by this hormone that increases during pregnancy. So we decide to pursue GPR1. First thing we have to do is validate that it actually works. So we make a clean knockout line. Control is in purple, the GPR knockout is in green. We treat them with interferon. The cells all turn green, but when we wash it out, the GPR knockouts maintain that signaling compared to control. That's quantified here. We can use pharmacological inhibitors of GPR. So this is 17-beta estradiol. This is its normal ligand. And there are compounds that look kind of like estrogen, but are specific for GPR1, don't hit the classical estrogen receptors. In this case, G15 is the antagonist. So binding of G15 prevents activation of GPR1 activity. When we add that drug in a dose-dependent manner, we stop cells from turning off their interferon signaling after removal of the cytokine. This is with the reporter. This is with endogenous ISGs. We add interferon, cells express the ISG to a slightly higher extent, just basally when GPR is inhibited, but then that response turns off much slower than the control-treated cells. So that is knockout and inhibition of GPR. Overexpression of GPR gives us the reciprocal phenotype, as one might expect. So again, with the reporter here, we've cloned the gene, we've overexpressed it. The control cells respond to interferon, the overexpression cells do not. This is an endogenous transcript for IFIT1, which again is just a representative ISG. There's no response when GPR is expressed. This is a Western blot showing at the protein level. I think you can appreciate here, before interferon treatment, there's no interferon stimulated gene expression occurring. But in control, at different time points after infection, you're getting expression of the genes. But in GPR overexpression, you just get no accumulation of interferon stimulated genes. So it seems like our screen is real, that GPR is a real hit. But we need it to be more than just an interferon suppressor. We need it to perform this differential interferon regulation, right? We need it to leave peripheral maternal tissues alone and specifically protect fetal tissues. So can it do that? So from a pregnant mouse, we took a number of tissue samples that I'm showing you here. And the two to draw your attention to is the lung, where we're using flu as a challenge model, or we will be. There's very little expression in the lung, but in the placenta and the fetus itself, there's high expression of GPR. So that leads us to this model. In the lung, there's low GPR. Presumably interferon signaling can occur even when estrogen is high. That helps control the virus in maternal tissues. At the same time, in the fetus and the developing placenta, GPR levels are high. That should suppress interferon signaling when GPR is activating ligand 17-beta estradiol is present. That should lead to protection of the fetus. So we have a good flu infection model during pregnancy, which allows us to test this. We mate the animals. At day 5 post-mating, we start giving this antagonist. So this is like mimicking a GPR knockout. We infect them with flu and we see what happens. Now we're looking first at the maternal side of things. And there's no difference, plus or minus the drug, in mock-infected animals. The animals are gaining weight because they're pregnant. They're giving birth here. There's no difference in weight gain or time to birth. The infected animals gain less weight because they're sick, but again, there's no difference between the blue and black lines here. If you look at viral titer in the lungs, again, the drug makes no difference, right? This is just looking at the maternal side of things. The virus or the receptor GPR is not expressed in the lungs. It makes sense. This is what we would see. You can even dive a little deeper and look at the transcriptional response to the virus in the lungs. This is just comparing infected and uninfected lungs with no drug. We get this kind of prototypical antiviral response. These are antiviral genes being turned on in the lungs. But if we take these same lungs and compare infected lungs plus and minus drug treatment, all those changes go away. The lungs are responding exactly the same, regardless of whether or not GPR1 is inhibited. The protein profile in the lung is unchanged similarly. And again, this makes sense. There's no receptor. Inhibiting that receptor doesn't matter, or at least there's lower levels of the receptor, and inhibiting it doesn't matter. But what about the placenta and the fetus where the expression of this receptor is high? This is similar to the experiment that I showed you before, comparing the placenta from an uninfected animal to an infected animal. You can see there's some gene expression changes. This is gene ontology of the upregulated genes. It's genes involved in immune activation, which we thought was interesting. The placenta is detecting the viral infection in the lung and causing there's some transcriptional responses associated with that. But now comparing placentas from infected animals plus and minus the drug, again, this is the condition that nothing changed in the lungs. Now we see significant gene expression changes. There's a number of genes upregulated and even more genes downregulated. This isn't because the virus now goes to the placenta. The virus is never detectable in the placenta. It's only detected in the lungs. So fundamentally, this normal response to maternal flu infection is changed when GPR is inhibited. It's not because the virus goes there. So what are these changes? As one might expect, the genes that go up are for the most part ISGs. The genes that go down, though, interestingly, if you perform an unbiased gene ontology analysis, the top 5 most significantly enriched pathways all have to do with blood vessel development. This is satisfying to us because interferon signaling is mutually exclusive with angiogenesis, so that makes sense. But the placenta, kind of its whole job is to have blood vessels, right? The placenta is the organ that is mediating nutrient exchange between maternal and fetal blood supplies. So it seems like this gene expression profile would be bad for the placenta and subsequently the developing fetus. And it turns out that that is indeed the case. So mice have this vascular labyrinth structure in the placenta. You want to see this open space. These are red blood cells, and this is where nutrient exchange between maternal and fetal blood is occurring. If GPR1 is inhibited and the pregnant dam is infected, you can see that this architecture is basically completely lost. Now, in terms of the pups that are born, there's really no difference across the different treatment groups. There's still pups born in all of them, but in terms of viability, if the pregnant dam is infected and GPR1 is inhibited, essentially all these pups are born non-viable. This is an example of what it looks like, and I want to walk you through this because this is a key point. GPR is dispensable unless maternal infection is occurring, if there's maternal inflammation. So the pups are all fine. These are viability numbers in the corner. The pups are all fine if GPR is functional and the pregnant dam is not infected. If GPR activity is suppressed with this drug G1, but the pregnant dam is not infected, the pups are all fine. GPR doesn't matter. There's no inflammation to suppress. Similarly, if the pregnant dam is infected, but GPR1 is there, the pups are all fine because GPR1 is doing its job. It's only when we suppress the ability of GPR to perform its function in the context of infection that this really matters. And this magnitude, this phenotype and kind of the magnitude of effect on the pups is similar to what we see in Zika models of mouse infection. So it seems like we've generated some data that supports this model, but what I haven't done is linked it to type 1 interferon. That's how we got into this whole thing. So the last experiment I'll show you is the same experimental diagram before, but now we've replaced flu with poly-IC, which is a viral RNA mimic. Because we're going to put in antibodies to block interferon signaling, we don't want to change the course of disease in maternal tissues, as would occur in the lungs if we suppressed interferon signaling. So now we're giving poly-IC instead of the virus. We're giving either control or an interferon blocking antibody with G15 treatment. We're collecting fetuses at E16. In the control, no drug, no poly-IC, no antibody, the fetuses are all fine. With a control antibody and poly-IC, we get some fetal reabsorption. If we add G15 to compromise JEEPR activity, things get worse. But now we can take this exact condition and block the interferon receptor, and all the pups are fine. That's quantified here, arguing that the G15 phenotype, the JEEPR1 inhibitory phenotype, is really acting predominantly via regulation of the interferon response in the placenta. So I'll wrap up with a model for the second part. We think early and in pregnancy, there's low expression of JEEPR here, there's high expression of JEEPR here, but estrogen is low. So interferon signaling is allowed in both places, which inflammation is actually important for the early stages of the establishment of pregnancy. As pregnancy proceeds and the placenta matures, it actually becomes the major source of estrogen itself. High local concentrations of estrogen combined with high expression of JEEPR leads to differential suppression of interferon signaling here. Interferon signaling can still occur if the mother is exposed to a pathogenic insult, whereas here the fetus is protected. So I'm out of time. I'll wrap up. Appropriate type 1 interferon signaling is context specific, which I think people understand, but I think it's worth making that point. Maybe a less kind of intuitive point that I want to make is that active regulatory mechanisms, in particular, negative regulatory mechanisms, are required to prevent inappropriate signaling. So you can't just turn off the induction side of things. You have to have a suppression pathway on the back end, which I think is less appreciated, and what both of our stories today are going to in terms of a point. We've identified ETV7 as an interferon stimulated gene itself that serves as a brake for other ISGs, and we think maybe linked to immunopathology in the lung during flu infection. We're continuing to work on that. JEEPR controls interferon signaling in a temporal and tissue-specific manner during pregnancy. And we think the reason that we don't know about JEEPR is because it's an invisible system, or that we didn't know about it before it works, right? So the idea was that congenital infections, or these infections that are fetal tropic, cause these issues because they go there, right? And anything that doesn't go there doesn't matter. But what we're saying here is, again, that potentially all maternal infections would have significant negative complications for fetal development, if not for these protective systems that are likely just overwhelmed when the fetus is replicating in those tissues. So these are the people who actually did the work. The ETV7 stuff in the lab was done primarily by Heather. It's a graduate student in the lab. Al was a graduate student. He recently defended and left to start a postdoc. He did the ETV7 work. We're funded by a number of great agencies. We've been lucky to work with them, and various collaborators. Peter, Jennifer, and Ben on initial survivor cell stuff. Jean, we did the JEEPR study with her, and we have ongoing collaborations with Ryan and Sarah. I will stop there, and I'd be happy to take any questions.
Great. Thank you so much. That was really phenomenal. Looks like people have their hands raised. So we can just unmute and people can ask questions if they want to chat, put it in the chat box. I'll also read that. Lee Fan, I think you have a question.
Yeah. Hi, Nick. That's fantastic talk. I really enjoy it. I have two questions. The first question is, what is the benefit to have those survival cells? You talk about they're bad. We should eliminate them, but what is the benefit of having them? The second question is, as you show that the JEEPR, in the JEEPR story, it helped so basically inhibit the type 1 interferon signaling. but you also said the Zika virus still killed them. I mean, the GPR wasn't inhibited in that study. So the reason that in Zika infection, it didn't work as well compared to the influenza infection, is it because GPR expression is just not enough and being overwhelmed by the concentration of type 1 interferon in the placenta, or the viral infection at the placenta negatively impact the expression of GPR1?
Yeah, both absolutely great questions. So for your first one, why have we evolved to have cells survive if they're bad for you? And you sound like reviewer 3 on my grants. We actually spent a lot of work to figure this out. And it turns out that inflammation that is made is detrimental at the resolution of the primary infection, the primary pneumonia. It's leading to more inflammation, which is bad for that aspect of the disease. But that inflammation also protects you from secondary infection for a period of about 2 to 3 weeks. So we're able to show with the depletion, although the animals heal faster, they're now more susceptible to secondary infection. So we think that evolutionarily, there's a trade-off, right? You don't want to hurt your lung is more than necessary, but we think that that's kind of balanced to some extent by protecting yourself from secondary infection right away. So I could show you the data if you want to see more, but we think that's the answer. You're willing to tolerate a little more inflammation at the resolution of the primary pneumonia to protect you from a secondary pneumonia. The question as to why doesn't GPR work for everything is another great question. And we don't know. We actually did some Zika virus experiments that I didn't show you. And GPR helps protect from Zika. So if you knock out GPR, Zika, at least in a mouse model, is worse than it would normally be. I favor your first hypothesis. That's just local concentrations of interferon are high when the virus is replicating in that particular tissue and just overwhelms what GPR is able to do. It would be really exciting if the virus had an active suppression mechanism for GPR. And that's something we've thought about and something that we're looking at now. It's kind of hard to tell because lots of stuff goes bad when the virus is infecting those tissues. It's hard to tell a difference between just tissues dying and falling apart and active reduction in gene expression. But we're working on that now as well.
Thank you.
Elena, you have a question?
Yeah, Nick, really, really nice talk, very engaging. Thank you so much. So I also have 2 questions. So the first one is the ETB7. So it suppresses ISGs by binding at the ISRE. And so my question is that, while it's suppressed, you think, or at least the way I understood the model, is that there must be something that is inducing these ISGs. And so, but you said, if I understood correctly, that there's no more interferon. I guess there's no viral RNA that persists, that triggers interferon. So I'm a little bit curious on what is inducing the ISGs for a long time, while ETB7 suppress them, but what is inducing them?
Yeah, that is a great question, and it's also one of the things that keeps us up at night, right? There's two options. The first is that maybe there is low level of interferon, right? And the virus is cleared to our ability to detect, but what if there's like 3 molecules of viral RNA in the cell, right? Like that's enough to be sensed and make interferon. So that's one possibility. It's just a really low level of interferon, and we can't detect virus, viral transcripts that are driving it. The other option is that it's just interferon independent expression of ISGs. And for sure, that occurs, right? And I didn't show you some of the data. We actually have some really nice epigenetic data from our survivor cells. And what we think is, so this is what I think is happening. So the short answer is we don't know for sure. It could be that there's interferon. What I think is what I'm going to tell you now. When these cells are infected, there's just, everything is going so poorly for the cell. All of these ISG promoters, you know, epigenetically, chromatin-wise, are wide open. The cell is trying to drive these processes as hard as it possibly can. And most cells die, so it doesn't matter. But if the cell comes through it, basically epigenetically, these promoters are still open, much more than an uninfected cell would be at the same loci. And so I think that it's probably interferon independent, just maintenance of transcription of these loci, because epigenetically there's been massive dysregulation. And that's what's causing ISG transcription. Why ETB7 doesn't appear to fall into this other group of ISGs that stay on, and why it gets turned off, that we have no idea. But
No,
that's
very
that's
interesting. Very super interesting. And then the second question is the GPR. So you saw that there was also high expression of GPR in other tissues, like I think, if I catch it correctly, it was the liver, for example. And so how these tissues are, you know, which are the implications for these infections in these other tissues in the context of pregnancies or estrogen supplementation? You know, you'd expect that they are more susceptible to infections. And have you tested ISGs in the other tissues that they express high GPR when you did your studies?
Yeah. So this again is a great question, and we don't know the biological threshold for meaningful GPR activation, right? So women have higher estrogen than men, right? For example. And there's hormone supplementation therapies and all these things that you alluded to. But where that takes you relative to pregnancy levels, I mean, it's still lower, and we don't know where that threshold is. There are some interesting immunological phenomena that are also reported in pregnancy. For example, women with rheumatoid arthritis during pregnancy frequently report alleviation of symptoms. GPR is highly expressed in hematopoietic cells. Like if I would have put the spleen on it, the spleen is probably the next highest outside of, right? So we had reproductive tissues like ovary and uterus. Next highest is probably the spleen, if I'm remembering correctly. And it would be really interesting if just during pregnancy, if, for example, rheumatoid arthritis was, you know, these macrophages or whatever else, you know, the GPR on them is responding to this estrogen. These are early days. These are early days in that project. So, you know, we want to expand into those areas. But one thing I maybe briefly made this point that is really exciting is that there are small molecules that are specific for GPR that don't target the other estrogen receptors. So if we're able to define these kind of therapeutic thresholds, one could potentially deliver GPR specific agonists to activate these processes without the other effects of kind of hormone that you would expect from super high levels of hormones like estrogen. And that may be an interesting, it may be an interesting way to like take the good aspects of this in terms of presenting disease or globally suppressing basal inflammation. I mean, all kinds of stuff is associated with too much inflammation, right? Like heart attacks and, you know, these types of things. It'd be really great if that was true. But we haven't formally tested it.
Yeah, terrific. Thank you so much.
Thank you.
Great. Are there any other questions? Well, if not, thanks, Nick, for a really, really fascinating talk, and thanks everybody for attending.
Yeah, no, my pleasure. Thanks again for the invitation.
Great talk.
Thanks, Nick.
Thank you.
Thanks, Jack, too.
So Nick, I think you're on at 11 with Alistair. You have the Zoom link and everything?
Yep, perfect.
Okay.
And then I'll chat with you after.
Yeah, great. All right.
All right.
Thanks so
Jack.
much. All right, take care.